Hey everyone, welcome back to Window Seat Studies. I hope you all are doing well. So, we were learning about cellular signaling, in which we have covered different cellular signaling pathways which work through the activation of receptor tyrosine kinases as well as GPCRs. Then we started discussing about the signaling pathways which needs the receptor proteins besides those receptor tyrosine kinases and GPCRs. So in last class, we learned about JAK-STAT signaling pathway, where the cytokine receptors they are being coupled with cytoplasmic tyrosine kinases, and they are going to do the downstream signaling. Today, we are going to learn about TGF-beta signaling, where the TGF-beta proteins, these are the extracellular signal proteins, and they are going to work through a receptor which has intrinsic serine threonine kinase activity, and it is going to activate SMAD protein downstream and going to result into the transcription of genes which are very important in during the development of cells during the developmental stages. It is important for pattern formation, and in adults it is important for tissue repair. So today we are going to discuss about TGF beta signaling, where the TGF beta receptor is a serine threonine kinase. Okay, so this TGF beta pathway, the TGF beta signaling pathway, it resembles a lot with JAK-STAT pathway. The way in the JAK-STAT pathway, the receptor, the cytokine receptor, it activates the kinase domain, which is going to phosphorylate a downstream protein, which is a latent transcription regulator. In the same way, in the TGF beta signaling, SMAD protein will be activated, which is also a latent transcription regulator. Means it is going to go once it is activated, it is going to translocate in the nucleus, and it is going to regulate the transcription of certain genes. Okay, so such factors, such proteins are known as latent transcription regulators. So in the case of JAK-STAT pathway, STAT proteins were the latent transcription regulators, and they were being activated by the receptors, cytokine receptors, which were being coupled with Janus kinases, right, JAKs. In this case, the TGF beta receptors, instead of having tyrosine kinase activity, so the JAK Janus kinase, it was a tyrosine kinase. But in this case, the TGF beta receptors, they are serine threonine kinases, means they are going to, serine threonine kinases are going to phosphorylate serine and threonine residues of the peptide of the protein. So let's start learning about the signaling pathway in detail. So this TGF beta family, this TGF beta superfamily is a large family. It is large family of proteins. Almost there exist 33 different types of TGF beta proteins in humans, and they are very important because this TGF beta signaling is going to result into different cellular responses, such as pattern formation. It is very important for pattern formation in developmental stages. Okay, it is also important, it also influences or has effect, so it influences cell proliferation, cell specification, cell differentiation, and it also affects extracellular matrix production and even cell death. This happens during the developmental stages. So during developmental stages, this TGF beta signaling is crucial for influencing cell proliferation, specification, differentiation, the extracellular matrix production, and even the cell death. And in the adult, the TGF beta signaling, it is involved in tissue repair. Okay, so it is involved in tissue repair. So this happens during the developmental stages, and this happens in the adult stage. Okay, so this is how TGF-beta signaling is very important in during the developmental stages from embryo to the next developmental stages, and even during the adult stage. So this TGF-beta signaling. Okay, so TGF-beta superfamily. So this TGF-beta superfamily, I told you that there exist multiple types of proteins in this superfamily. So it is categorized into two subfamilies. Okay, so TGF-beta, one subfamily is known as activin family or activin TGF-beta family. Okay, so TGF-beta superfamily is categorized into two. And the other family of the TGF-beta, which belongs to TGF-beta superfamily, is BMP family, bone morphogenetic protein family. So those two families, they are the part of TGF-beta superfamily. So they act, so activin molecules, TGF-beta molecules or BMP molecules, they act as the extracellular signal. So they are usually the homodimers, okay, and they are going, so these are the, so TGF-beta superfamily has dimeric extracellular, so these are dimeric extracellular signal molecules. Okay, so those extracellular signal molecules, they are received by receptors. So as the TGF-beta is the protein which is being acting as the extracellular signal. The receptor is also referred as TGF-beta receptor. So this TGF-beta receptor has the serine threonine kinase activity on the cytosolic part. So in the intracellular domain, it has the serine threonine kinase domain. So STK activity is present on the cytosolic side, which is going to do the downstream job. Okay, so I introduced you about all those stuff. So let's now start having a look at the signaling pathway. So let's say that this is the plasma membrane, okay, over which the TGF-beta receptor resides. So TGF-beta receptor, there are two types of TGF-beta receptors that are present in the cell. First is the type 1 receptor and other is the type 2 receptor. Okay, so these two types are different. They are functionally different and they are structurally different as well. And both of them, they exist in the homodimeric form on the cell surface. Okay, so as we learned in RTKs, the RTK receptors, they were simply present on the cell surface receptor as the monomers, and due to the arrival of extracellular signal, they used to get dimerized, right? But in the case of TGF-beta receptors, they are already present in homodimers. And there exist two types of receptors, type 1 and type 2. So let's say that this is the plasma membrane over which resides the TGF-beta receptor type 1. So this is the type 1 receptor. Okay, so this is the homodimer of type 1 receptors, and this is the type 2 receptor. This is the homodimer of type 2 receptor. Okay, so both of them, they exist in dimerized form, and the TGF beta molecule is also a dimer. So this is TGF beta molecule, which is going to act as a signal, and it is going to result into the tetramerization of this tetramerization of those receptors. Why? Because they are already present in dimerized form. So type 1 is also a dimer and type 2 is also a dimer. Both of them, they are homodimers. And when the TGF beta comes, it arrives on this on the site. When it interacts with those type 1 and type 2 receptor, it results into tetramerization of those receptors. So what will happen? So on the cell surface, okay, so this is the plasma membrane. Okay, so the type 1 and type 2 receptors, they will get tetramerized. Okay, and this is the TGF beta molecule which has been bound to the receptors. So this will bring the serine threonine kinase domains of both of the proteins together. So the intracellular part has the serine threonine kinase domain. So this is a serine threonine kinase domain, and this is also serine threonine kinase domain. So due to the tetramerization, the serine threonine kinase domains of the both of the proteins, they are going to come in the close proximity. So the physical proximity of the serine threonine kinase domains will activate them. Okay, so what will happen that the T2 will get activated and it is going to result into phosphorylation of T1 domains, the T1 intracellular domain. So what will happen? T2 will phosphorylate, it will phosphorylate T1. So T2 will phosphorylate T1. So this is a different case from RT case. In RT case, both of the receptor domain, they are going to phosphorylate each other, resulting into trans autophosphorylation. But in the case of TGF beta signaling, the TGF beta receptors, the type 2 receptors will get activated and they will phosphorylate type 1 receptors. Okay, so type 2 will not get phosphorylated. It is going to the receptor titers, the serine threonine kinase domain of type 2 is going to phosphorylate type 1. So now the phosphate group will be added on the type 1 residue. Okay, so here we see the phosphorylation of type 1 residue. So this will result into the activation of type 1 receptor, right? So to this phosphorylated serine threonine kinase residue, a protein will come and bind, and that protein is a latent transcription regulator, and it is referred as SMAD. So to this phosphorylated domain, so to the phosphorylated type 1 receptors, SMAD proteins will come and bind. Okay, so this will result into a conformation, something like this. So to this phosphorylated residue, a SMAD protein is binding. Okay, so let's assume that this is a SMAD protein. So SMAD protein is now bound to the phosphorylated receptor tyrosine kinase residue. No, sorry, not the tyrosine kinase, the serine threonine kinase. So now let's assume that this is SMAD proteins. So this SMAD protein will get attached to the receptor and this will result into the phosphorylation of SMAD protein. Okay, so once the SMAD protein binds to the receptor, the intracellular domain of receptor, it will get phosphorylated by type 1 receptor. So type 1 receptor, so the serine threonine kinase domain STK of type 1 receptor, it is going to phosphorylate SMAD, and this phosphorylation of SMAD protein will release the SMAD. So this phosphorylated SMAD will be released in the cytoplasm. Okay, and that is going to now form a trimeric complex. So this SMAD, this phosphorylated SMAD is now referred as R SMAD. Why? Because it is receptor activated SMAD. There exist different types of the SMAD proteins, R SMAD, co-SMAD. We'll list them down. Uh, we'll list them down in a while. So the R SMAD are the receptor activated SMAD proteins, which will get now phosphorylated. So once they are phosphorylated, they will be released in the cytosol, and this R SMAD will form a trimeric complex. So two R SMAD, they will bind with one co-SMAD protein. Okay, that is going to result into a trimeric complex. This trimeric complex, this will move, this will translocate in the nucleus. and that is going to do the downstream job. So it is going to switch on certain genes and going to result into the transcription of certain genes. Okay, so the SMAD proteins, there are different types of SMAD proteins. Okay, the R SMADs and the co-SMADs. So R SMAD proteins are the ones which are going to bind to the receptors. Okay, there exists one more category, R SMAD, co-SMAD and inhibitor SMADs. These are different types of SMAD proteins. So the R SMADs are the ones which are going to bind to the activated receptor. So these are the receptor activated SMADs. Okay, they are also categorized into two different categories depending upon which type of TGF beta protein is going to activate them. So let's say that activin molecule, so activin receptors are the ones which are going to activate our SMAD. Then those SMAD proteins are usually SMAD2 or SMAD3. So SMAD2 and SMAD3 proteins, they get activated or they get bind to the activin receptors. Okay, while the BMP receptors, they can activate SMAD1, SMAD5 and SMAD8. Okay, so those different types of SMADs, so SMAD 2, 3, 1, so SMAD 1, 2, 3, 5, 8, all of them, they are R SMADs, means they are going to bind to the activated receptor. They are going to bind to the activated TGF beta receptor and they will form trimeric complex with co-SMAD, which is also SMAD4. Okay, so they are going to form a trimeric complex with SMAD4 protein, and depending upon which SMAD protein forms the trimeric complex, depending upon this point, the downstream gene, the target gene will be decided, and that target gene will get activated. Then there exist inhibitor SMADs, which are which will inhibit the signaling pathway. That is why they are referred as inhibitor SMADs. So the inhibitor SMADs are SMAD6 and Smad7. So they are going to use the negative feedback loop to inhibit the signaling pathway. We'll also learn about how the inhibitors SMAD do their job. So right now what we want to focus is that SMAD2 and SMAD3, SMAD1, 5, 8, all of those are the R SMADs and SMAD4 is the co-SMAD. So the R SMAD, two R SMADs and one co-SMAD, they are going to form a trimeric complex which will activate or which will function as a transcription regulator for the target genes. Okay, so this is the basic signaling pathway, and this signaling pathway is inhibited by a negative feedback loop. So we have been discussing about we have discussed about negative feedback loop multiple times. So just for your reference, negative feedback loop is referred to a loop when certain protein, it inhibits its own production when the concentration of that protein increases. So let's say that due to certain signal, production of protein A is seen. Okay, so this protein A, once it increases in its concentration, it is going to do some, it is going to recruit some inhibitor proteins, or it is going to somehow inhibit the transcription of the gene which was initially coding for protein A, and this loop is known as negative loop. Means this protein A, when the concentration of protein A increases inside the cell, that is going to inhibit its own formation. That is what we refer as the negative loop. So in the case of SMAD proteins, there exists inhibitory SMAD which will get, so what will happen, the co-SMAD and R SMAD trimeric complex, this trimeric complex will switch on some genes, so it is going to result into transcription of some genes, right? So the group of gene it activates, in that group of gene it will also activate some inhibitor SMAD genes, and those inhibitor SMAD genes will produce SMAD6 and SMAD7, which is going to now inhibit the formation of this trimeric complex so that the transcription of target genes will be stopped. And this is what we refer as the negative loop. So how does SMAD6 and SMAD7 do this job? So how does they inhibit the transcription of the formation of complex R-SMAD and co-SMAD? So what do? They have three, no, they have four choices. Either they can, they can compete with R-SMAD. So what do? The SMAD6 and SMAD7, they compete with R-SMAD so that R-SMAD will not bind to the receptor protein and it won't get phosphorylated. So now the R-SMAD is not phosphorylated, the trimeric complex will not form, and that is going to result into the disruption of the signaling pathway, right? Or they can bind to co-SMAD. So either binding with R-SMAD or co-SMAD will result into the so this will avoid the formation of the trimeric complex, and that is going to result into the switching of the Wool signaling pathway, right? If the trimeric complex is not formed, it is not going to result into the activation of downstream genes, and that is why the Wool signaling pathway will get disrupted. So the third way that this SMAD6 and SMAD7, the inhibitor protein works, is that they recruit SMURF, SMURF. So SMURF is a SMAD ubiquitination and regulatory factor. So this is SMAD ubiquitination and regulatory factor. So recruitment of SMURF will result into the ubiquitination of the TGF-beta receptor, because this is the ubiquitination regulatory factor, right? So this SMURF, which is recruited by SMAD6 and SMAD7, this will result into ubiquitination of TGF-beta receptor, and we have already discussed this in the post-translational modification series, that ubiquitination, this modification tag refers to the to the protein degradation. So this is also known as protein degradation tag. So if any protein undergoes ubiquitination, this means that that protein is meant to be degraded now. So it is a tag, like this is a vest. So like when you organize your room, you take out certain books and label them as those books are to be donated, right? So when you take them out and put them in certain box, so in the donation box, that is so that puts a label on those books that they are meant to be donated now, right? So you organize them and you separate them inside certain box which has been labeled as a donation box, right? So in the same way, inside the cell, when ubiquitination, this tag, this chemical modification is being added to any protein, the cell is actually labeling that protein, that this protein is not this is not useful now, and this protein should be degraded. So this will result into internalized. So now the TGF-beta receptor has been ubiquitinated, means the cell has put the label of useless protein over that protein. So this means that this TGF-beta receptor will be internalized and it will be degraded. So it will be internalized in the endosomes and it will be degraded. So this will result into the degradation of whole receptor. If there is no receptor, then the whole pathway will degrade. So this is how SMAD6 and SMAD7, they will either compete with R-SMAD or co-SMAD. They will bind with them and they will inhibit formation of trimeric complex. So trimeric complex will not form, or they can directly recruit SMURF so that the whole receptor protein is degraded. So that even if there is extracellular signal in ample amount, if the receptor protein doesn't exist on the cell, it is not going to result into the signaling, into the start of the signaling pathway. The fourth way by which the SMAD6 and SMAD7 work, that they recruit certain phosphatases. So they recruit phosphatases. So those protein phosphatases, they will for dephosphorylated the phosphorylated Smads, or they will dephosphorylated this phosphorylated TGF-beta receptors. So this will result into the disruption of cell signaling. So so these are the four different choices that inhibitor proteins have, and it will choose either of them so that the TGF-beta signaling will be stopped. And this is how they are going to inhibit whole signaling pathway using a negative feedback loop. So whenever our co-SMAD and our SMAD, they are going to transcribe the downstream genes, they are going to transcribe the target genes, they will also transcribe inhibitory SMAD after a certain point. That is going to result into the disruption of the signaling pathway using those four different choices, either of those choices. Okay, so this is how the whole pathway is disrupted. So with this, I conclude the lecture. But before concluding the lecture, let's have a quick summary of the whole lecture. Okay, so here is the whole signaling pathway. The schematic belongs to the Bruce Alberts, and this single schematic explains the whole pathway. So here you can see that the TGF-beta receptors, they are present into already dimerized form. So this is the type 2 TGF-beta receptor, and this is type 1 TGF-beta receptor. So the TGF-beta, which is dimer, it comes and it binds to those type 1 and type 2 receptor. So it brings the TGF-beta receptors together and it results into the tetramerization. Okay, so a tetramer of the TGF-beta receptors is formed. So these are the TGF-beta receptors, right? So a tetramer will be formed, which will result into the physical proximity of those serine threonine kinase domains. So the TGF-beta receptor has serine threonine kinase domain. So now the type 2, here you can see that this is the, this is the type 2, this is the type 2 receptor. Okay, that is going to get activated, and the activation of type 2 receptor will result into formation of type 1 receptor. So type 2 is going to phosphorylate type 1 receptors. Okay, so this is not the transphosphorylation event. This is very different. That only one serine threonine kinase, only one type of receptor is activating its serine threonine kinase domain, and it is phosphorylating other type. Okay, so type 2 phosphorylates type 1 receptors. So once those type 1 receptors are phosphorylated, they get activated. Okay, and this activation will result into phosphorylation of SMAD proteins, R SMAD, which are present abundantly in the cytosol. Okay, so SMAD protein will get phosphorylated, and that will change its conformation, opening its binding site for the other SMAD protein. So this phosphorylation will result into the opening of the binding site of SMAD protein. Okay, so you can see here, so the binding sites which were, here the binding sites were hidden, and due to the phosphorylation, the binding sites, they are now open, and to those binding site SMAD protein can go and bind. So two R SMAD will bind with one co-SMAD, which is SMAD4, and that is going to form a trimeric complex. So this trimeric complex will now translocate into the nucleus, and with some other transcription regulatory factors, it is going to form a transcription regulatory complex, which will bind to the DNA at the TGF-beta responsive cis regulatory sequence, and it is going to do the transcription of the target gene. Okay, so this is particular element to which this TGF-beta, no, the SMAD complex, the SMAD regulatory complex, the SMAD transcription regulatory complex binds, that element in the DNA is known as TGF-beta responsive regulatory sequence, because this sequence is doing the regulation of certain target genes, and that is responding, or it is working only when the TGF-beta is present in the biological system. That is why it is referred as TGF-beta responsive. So it is responding to TGF-beta, and that is why it is called TGF-beta responsive cis regulatory sequence. Cis because it works on the site. Okay, so it is present on the DNA itself. That is why it is referred as the TGF-beta responsive cis regulatory sequence. And activation of this regulatory sequence will result into the transcription of the target genes. So when the target genes are transcribed, what will happen? So once the target genes are transcribed, with the target genes transcription, the SMAD, inhibitory SMAD, SMAD6 and SMAD7, they will also get transcribed, and they are going to now inhibit either they will directly degrade the type 1 or type 2 receptors by recruiting SMERF, okay, or they can inhibit the formation of this trimeric complex by binding to either R-SMAD or co-SMADs, or they can recruit phosphatases which will result into the dephosphorylation of phosphorylated receptors or dephosphorylation of phosphorylated SMADs. So either of those pathway will be chosen by the SMAD6 and SMAD7 inhibitory SMAD proteins, so that the whole pathway, this whole TGF beta signaling pathway disrupts. So they are only produced, they are only transcribed when the downstream, so when they will be only transcribed when the SMAD proteins, they are going to, they are going to result into formation of target gene. So that is why this feedback loop is known as negative feedback loop, because the pathway which was activating the target gene, the same pathway, once it is activated, it is deactivating its own itself, right? So the pathway which activates and produces certain protein, that protein itself deactivates its own production pathway, and that is why this loop is known as negative feedback loop. So this is how the inhibitor proteins which are produced due to the negative feedback loop, they will disrupt this whole signaling. So we are done with TGF beta signaling. In next lecture, we'll learn about Wnt signaling, the Notch delta signaling. So all of those signaling pathways, they are very important, because they are not, we will learn them here in cell biology series, and they will be very important and they will be very useful when we will start learning about developmental biology. In developmental biology, I won't be referring to any of those, any of the details, mechanical details of those pathway, how those pathway work. I'll only address that, okay, so for this pattern formation, TGF beta signaling is required. For this particular event, Wnt signaling is required. So when I refer to that, you should remember all those details of the pathway, how the pathway gets activated, and how the pathway will be inhibited. Okay, so I conclude the lecture here. In next lecture, we'll discuss about Wnt signaling or Notch delta signaling. Till then, see you. Bye bye.